My name is Eve Tesiudi and I'm in the group of Jörg Stelling at ETH Zurich, and together with Javier Santos Morenos we worked on a robustness and innovation in synthetic genotype networks. When we discuss the phenotype of an organism, we're essentially referring to the specific characteristics it possesses. This phenotype, for example, the patterns in this butterfly, is determined by the genetic material, the underlying genotype of the organism. Let's take an example where the genome of an organism specifies the interaction of three genes with one another. This interaction can lead to the creation of three distinct colors, for example here blue, green and orange, that when combined form a spatial pattern. In the process of evolution, it is necessary for genotypes to generate new phenotypes through mutations. However, if the majority of the mutations lead to entirely new phenotypes, preserving successful phenotypes would become exceedingly complex. Therefore, evolution must simultaneously be conservative, safeguarding functional phenotypes, and innovative, discovering intriguing new phenotypes. But how can these seemingly contradictory strategies be harmonized? The solution lies in genotype networks. A genotype network refers to a concept that encompasses a collection of genotypes that share two key characteristics. Firstly, these genotypes produce the same phenotype. Secondly, they are interconnected through individual mutation steps. Genotype networks enable evolution to maintain, on the one hand, conservatism by allowing genotypes to undergo mutations while retaining the original phenotype. Additionally, genotype networks can expand extensively across genotype space, meaning that significantly different genotypes can be reached through single mutation steps without losing the initial phenotype. Consequently, genotype networks promote innovation, as two widely separated genotypes that generate the same phenotype may only require one mutation to yield two distinct phenotypes. In this way, genotype networks offer both robustness and evolvability to the process of evolution. Until now, we had experimental proof of genotype networks existing in genotypes such as proteins or RNA. However, we lacked direct evidence of genotype networks in gene circuits. And now Javier will explain to us how we addressed this gap.
What we did is that we performed experiments using synthetic gene circuits based on CRISPR interference, and this provided us with the evidence that we needed. We combined those experimental data with mathematical models, actually up to 2 million mathematical models, and this allowed us to identify three different genotype networks. Two of them were providing spatial patterns, and then the third one was providing a different pattern, a temporal pattern, which is actually an oscillatory behavior. Now, within each of the genotype networks, we saw that there was a lot of interconnection between the different circuits, meaning that in face of potential mutations, those circuits would be robust and they would preserve the phenotype. Now, we also saw that there was interconnection between the different genotype networks, which means that some mutations would allow circuits to transition to another one that produces a new phenotype. This comprehensive approach allowed us to gain deep insight into the underlying mechanisms. So in essence, our research illustrates how evolution of gene circuits within genotype networks may be a key driver of evolutionary innovation.